Hi all, I am Md.Ahasan. I am going to present my project work, which is autonomous oscillating PDMS PCR chip for detection of nucleic acid analysis by using temperature gradient. Actually, it is the combination of two paper. One is oscillator, another is PCR chip. Here I presented the reference of to this paper. One is published in the Nature Communication, which is published by our professor. The name of the paper was multiple independent autonomous hydraulic oscillator, which was driven by constant pressure head. And another paper is published recently. So it is the oscillating flow microfluidic PCR chip, which use thermal gradient for nucleic acid analysis. So here, oscillating flow is produced by pump. So this is the main thing I described by highlighting. Offline detection is offline detection was the previous oscillating device which produce offline detection, but uh, now previous oscillating flow device require offline product detection. It cannot analysis live quantitative analysis by lively or vividly. So here I recommend a constant pressure oscillator which will produce constant pressure, which will produce oscillating pressure from constant pressure head and external PCR system uh, uh, for detection of nucleic acid analysis, where the combined annealing and extension is carried on by 56 degrees centigrade, and denature can be done by 90 degrees centigrade. By using this device, we can easily determine the fluorescence intensity emits from the after amplification of viral DNA or viral RNA by using proper PCR mixture. And we can also count the cycle number or melt analysis, which can be done with the experiment. So predetermination of cycle number and PCR mixture is not necessary here for detection. Again, only heater and image acquisition system is enough for this, and which will whole system take 30 minutes only to finish this experiment. The main motivation of this work was the feeding pump, which can be controlled by programmably from computer software. It can be eliminated by using autonomous oscillator, which can produce constant, which can produce oscillating pressure from constant pressure source. So this is the overview of this device. The figure 1A indicates the oscillator, and the inside figure below of 1A indicate the overall structure of oscillator, which consists of four important things: two valves, two capacitor, two reference resistor, uh, and two flow resistors, and finally reference pressure. Oh, actually, it is five important things. Sorry, I mentioned four. Again, the right hand side of this device, figure 1A at right hand side, which consists of some things like tape, In the figure 1b, it mentioned tape plus substrate and top and bottom, 2 inlet port, again a PDMS channel, which is PDMS channel of certain dimension. So all of them are used as an integrated device here. So we can easily use for the PCR reaction. This is the working principle of the oscillator. So input pressure is Pi, and if this valve opens at left side, then the pressure at top of the capacitor is become from minus output pressure P naught to Pi. At the same time, the bottom side of the capacitor will be energized because the top side pressure of the capacitor become Pi, and the bottom side pressure now it is gradually increased. So the fluid will flow from left to right, and it will go toward the valve V2, and it try to increase the gate pressure of the valve V2 and close this valve. Again, rest of the fluid will go to the reference because reference pressure is 0 kilopascal, which is significantly lower than input pressure Pi. So after that, the reference pressure is 0, and as valve 2 is closed, so in the right side capacitor, top side pressure should be equal to P naught. That means P output. This is minus 9 kilopascal. So the capacitor will deflect upward and create negative pressure at the bottom side also. So it will bring fluid from reference pressure toward this capacitor. So the fluid will flow from 0 kilopascal to capacitor. In the meantime, it will try to close the valve V1 as there is the motion of fluid. So in the meantime, the yellow channel indicate, previously I told that this is yellow channel indicates bottom of this device, bottom layer. So gradually the left side valve try to close, and the pressure gradually decreases. In the meantime, the valve 2 pressure was enough to open. So at that time, at 3 kilopascal, when it reaches the valve threshold pressure, then the valve 2 open, and this phenomenon, these things happen again cyclically. At figure 1b, it indicates that P naught is minus 10 and P in equal to 10. So it is oscillating between of them. And figure 1c, which is the integrated device. So basically, we can see from the figure D is not mentioned here. At figure D, you see that PCR chamber resistance is calculated almost 10 to the power 10. So it is not directly attached with the oscillator. Between of these two, there is a capacitor, so which block the flow from oscillator to PCR reaction happening channel, so that no fluid can go from oscillator to external device. It is ensured by using a capacitor C3 at figure D. So at figure C, I come back again. Fluid will move from bottom to top, top to bottom. It will be one cycle. So when the cycle number will increase like that, then we can see that the oscillation, for the oscillation, it will go up and down, and the cycle number will increase. So fluorescence intensity will increase here, and at the same time, we can see that if the relationship between temperature and fluorescence here, as you know that there is a temperature gradient here. So if the temperature will changes from top to left or top to bottom, then fluorescent intensity at higher temperature will decrease. And at the same time, and if we consider the gradient, we can see easily the significant change of the melting curve. So this is the application of result of this current work. So they use viral DNA to amplify and see the fluorescence changes. We can see from figure 1 to figure 1a, from cycle number 1 to cycle number 25, we can see easily that the intensity of fluorescence will increase, because we know at a higher cycle number, the number of copy of DNA will increase. So fluorescent intensity will also increase. Again, from the top to bottom, if we go from 55 degree to 80 degree, we can see the changes of at cooling time, at cooling condition, the intensity is higher than the heating condition. So we can summarize in figure B and C here. Fluorescence intensity will gradually decrease from 78 to 80 seconds. So it is clearly indicated in the figure C that clear change has been seen easily. So we can consider the melting curve here. Melting curve, I mentioned here, melting curve actually gives the result from the double stranded DNA to single stranded DNA at certain temperature, which will melt. So this will shows the melting temperature, because to do PCR reaction, denature happen. At denature, must be DNA, double stranded DNA must be separated into single strand DNA. So I propose a new method to do this. In the current design, it indicates the dimension of the channel. I try to make same length at 31.5 instead of I use 34.5 millimeter, but I changes some dimension of the width of the channel and implement height. As this paper I read it and I did not find any height, so I proposed height 50 micrometer and the width should be 100 micrometer. Then the resistance will be 10 to the power 13, which can make sorry, here I may written wrong, 10 to the power 10, which will produce 10 microliter per minute flow through the PCR device. Another important change I need to follow. We know the DNA for the amplification of DNA, 3 important time. One is denature, another is annealing, another is extension. So denature normally happened at 90 degree, extension at 72 degree, and annealing is normally 52 degree. But sometimes annealing and extension can be combined together for the simpler device, and it also work. In this case, I propose annealing and extension time as 28 seconds. So according to thumb rule, we know we can use 50 base pair DNA, double strand DNA, 50 base pair, so that we can easily broke the DNA hydrogen bond. And so at the denature time is 10 seconds. So to combine them from double stranded DNA to single stranded DNA, we need 30 seconds for annealing. This important things here, there is 2 different type of period. One is bigger than one, another is smaller. I can manage it by changing the oscillator periodic register. That means we can change the duty cycle by changing the period of oscillator. How I can change it? I change it by changing the periodic register left and right with 2 different value. For this reason, one half when pressure falls is 30 seconds, and one half when pressure increase is 10 seconds. By this way, I design the oscillator, which I described at figure 2, previous figure. So finally, Thank you for listening my presentation. And if any questions arise, please ask me